안녕하세요. Good day, everyone. Firstly, I'm so sorry that I can't speak well in Korean, so I have to conduct this webinar in English. I hope everyone can understand. Okay, I will try to speak slowly so that everyone can follow my content. So it's a great pleasure to be invited to this webinar. And my name is Edward, and I am the Regional Product Manager from Jinsscript. In today's presentation, I am going to discuss about the mRNA and also its applications, as well as to demonstrate how Jinsscript can help you in the mRNA production. So let me walk you through to the contents of my presentation today. I will begin with an overview of our company, followed by the introductions of the mRNA, along with some of the case studies that we have been conducted internally. And also, lastly, I will wrap up my presentations by illustrating our mRNA service and also other services that offered by Jinsscript. So Jinsscript was founded in 2002 in New Jersey, and we are also a service provider that well known in gene synthesis. In 2004, Jinsscript established the Research and Production Center in Nanjing, China. And throughout the years, Jinsscript has achieved numerous milestones, including the launching of the best zyme in 2013 in order to provide the industrial grade of the synthetic products service like enzymes, and also the Legend Biotech in 2014 for cell therapy platform. And also during the COVID-19 outbreak, Jinsscript collaborated with one of the professors from Duke NUS in Singapore to establish a neutralization antibody detection kit known as a CPAS. So in 2022, Jinsscript established a manufacturing facility in Singapore in order to serve the Asia-Pacific customer better. So based on the core gene synthesis technology and also other technologies, Jinsscript established 4 major platforms as you can see from the screen here, including the life science services and products in order to provide a one-stop solutions to the research community, a biologic CDMO platforms, and industrial synthetic biology products, which is known as best zyme that I mentioned earlier, and also the Legend Biotech for cell therapy platform. So now let's move on to the introductions about mRNA. So the messenger RNA, or known as the mRNA vaccines, have been studied for decades. But only recently, during the COVID-19 outbreak, this technology has caught everyone's attention. In contrast to the traditional vaccines, mRNA vaccine offers a more balanced immune response, triggering both the humoral and cellular components of the adaptive immune system. So let's get started to understand what is mRNA. I believe many of you are very familiar with the mechanisms of transcriptions and translation to the convert of DNA to mRNA and subsequently the proteins. So in this slide, I would like to mainly highlight about the mRNA transcriptions in the cell. As mentioned, the RNA is produced from DNA and after the transcription process. When an RNA transcript, it is first made in the eukaryotic cells, it's considered as a pre-mRNA and must be processed into the messenger RNA or mRNA. So a 5' cap is added to the beginning of the RNA transcript, and a 3' poly A tail is added to the end. So during the splicing, some of the sections of the RNA transcripts or the introns will be removed, and the remaining sections, such as the exons, will then be ligated back together again. So some genes can be alternatively spliced, leading to the productions of a different matured RNA molecules from the same initial transcript. So after it gets to the cytoplasms, the mRNA would have actually gone into different cycles. One of the cycles illustrates that the mRNA might be degraded, of which the poly A tail will be degraded by the enzymes known as the DA enzymes, and also the cap will also be degraded by another type of the unique enzyme. And in another pathway, the ribosome will bring together correctly the mRNA which binds bring together correctly to the mRNA, which needs to be translated into the by the tRNA, which will then assist in the translation. So when you zoom in this picture here, so during the initiations, most of the eukaryotic mRNA are translated by the cap-dependent mechanism, which requires the recognition by the EIF4E complex with the EIF4G and also the EIF4A. So this is so-called the EIF4F complex of the cap structures at the 5' end. And then the poly-A binding proteins, which is known as the PABP, will bind both the poly-A tail to this complex in order to promoting mRNA circularization. So the elongation is then assisted by the elongation factors such as the EIF4A and also the EIF2. And during this step, the amino acyl tRNA synthetase will catalyze the binding of the amino acid to the cognate tRNA. So you can see here, this is the picture that's taken by the electron microscope. The mRNA is this thin line, which is indicated here. And then the ribosomes are actually shown as the dark dot color. And then with this twisted line is in this picture indicating the elongated amino acid that actually forms the nascent proteins as shown in this picture. So this timeline demonstrated the history of mRNA technology development. So the mRNA was actually discovered since the 1960s. The characterizations of the mRNA and its technology has been studied extensively since then. But only until the recent years, this technology has been used for therapeutic purpose as a result of several challenges. So one of the challenges was that the mRNA can induce the immunogenicity, and it took scientists quite a long period of time to overcome this challenge. And in the year of 2008, the study had shown that the modifications of the nucleosides help to improve the RNA stability and the translations by preventing the signaling proteins within the cell from recognition and binding to the mRNA, so that it can actually minimize the immunogenicity. And another highlight here of this technology was the use of mRNA for vaccination purpose. So before the use of mRNA as a vaccine for COVID-19, this technology had actually been studied extensively as a vector for cancer research. In fact, in the year of 2012, the mRNA was being used for the first human test for personalized medicine purpose. And now let's move on and take a look on how mRNA is being engineered in order to improve the efficiency and the stability. So as you can see from this table, it demonstrated the elements that are usually being modified. First is the 5' cap, of which the capping can help to stabilize the mRNA and then to increase the protein translations. So this includes the cap 0, which is essential for the efficient translations of the mRNA, and then the cap 1, which is to evade the cellular innate immune response in vivo. And the addition of this 5' cap can be achieved through either the co-transcription approach or the enzymatic reactions approach. Second is the regulatory elements in the 5' UTR regions in order to increase the translations initiation. We usually will include the T7 promoter in these regions and the codon optimizations of the open reading frame regions in order to modulate the translation yield and also the nucleoside modifications. So in here is where the base can be modified, and we can substitute the uridine with the other pseudo-uridines in order to decrease the innate immune activations and also to increase the translations. Furthermore, the modulations in the 3' UTR regions will increase the intracellular stability. Additionally, it includes the microRNA binding site into the 3' UTR for tissue targeted translation. And finally, the inclusions of the poly-A tails over maybe 100 nucleotides or even 120 nucleotides to enhance the ribosome engagement and protect the mRNA from attack by intracellular enzymes. So with the poly-A tail, it can then help to prolong the mRNA translations and also the stability in cell. Now, let's talk about some of the mRNA delivery. So the IVT, or known as the in vitro transcriptions mRNA, has to avoid the degradations from the extracellular degradation agents, such as the ribonucleases, and also to interact the target cells in order to cross the cytoplasmic membrane and diffuse in the cytoplasm to reach the ribosome. So the formulations of the in vitro transcriptions mRNA in a suitable nanosystem or some of the vectors is also frequently a requirement for surpassing all of these barriers that I mentioned. So the non-viral vectors and more specifically lipidic system are the most studied among the delivery system to date. Currently, there are 4 major delivery methods for the mRNA as I've shown here. So commonly used delivery methods include the lipid-based systems, which is a cationic lipid that are able to interact with the mRNA by the electrostatic interactions, leading to the formulations of a complex called lipoplex. And the cationic lipids can then formulated to prepare the cationic nanoemulsions and lipid nanoparticles. LNP is also or known as or also include the liposomes and other lipid-based nanoparticles, and they are regarded as one of the most developed system to date for the mRNA delivery. So currently, there are several LNP platforms are at the forefront at the clinical trials as well. So as more data on the COVID-19 vaccines become available, studies have been conducted in a more systematic way to investigate the vaccine efficacy and to estimate the effectiveness of all of these vaccines against infections. And among the vaccines, the mRNA type of the vaccines from the Moderna or the Pfizer demonstrate an average of about 95% of the effectiveness against the COVID-19. So on the other hand, another types of the vaccine, which is a China-based biopharmaceutical company, which is known as the Sinovac, is behind this is the developers of this CoronaVac, which is the use of an inactivated vaccines, and it works by using killing viral particles to expose the body's immune system to the virus without the risking of a serious disease response. However, as you can see, compared to the Moderna and Pfizer's, the effectiveness of this kind of the vaccine is actually lower than the mRNA types. So this actually inspired the researchers to move on to the mRNA research for vaccine development to date. Okay, so another potential applications of the mRNA vaccines that I mentioned is to develop the mRNA vaccines for personal anti-tumor mRNA vaccines in order to express the neoantigens. So it is now clear that the neoantigen specific T cells are present in most of the cancers. And the neoantigens derived from the somatic mutations offers a specific and highly immunogenic target for vaccinations. And the recent developments of a rapid and also a relatively inexpensive technology for DNA sequencing has facilitated the identifications of these kind of targets. So from here, you can also tell that in addition to the mRNA vaccines against the infectious diseases, the mRNA vaccine can against the cancer cell is one of the potentials by its applications. So with that, I actually draw into a conclusions to demonstrate further and dive deeper with the applications. So in here, we are looking for major applications for using the mRNA, including the prophylactic vaccines or infectious disease vaccines, the cancer vaccines for the cancer immunotherapy, the proteins replacement, and also for gene editing therapeutic applications. In infectious disease vaccines, it requires the mRNA to encode the virus antigens. And as we all know that the classic example is the mRNA vaccines of the two approved COVID-19 mRNA vaccines from the Pfizer, BioNTech, and also the Moderna. In cancer vaccines, the mRNA will be encode the cancer antigens, including the common antigens or the personalized neoantigens. The mRNA can either be directly injected in vivo or transfected ex vivo for adoptive cell therapy purpose. For the proteins replacement, mRNA will be encoded for the proteins that are not expressed or are not functional, as well as the foreign proteins that activate or inhibit the cellular pathway. So these applications require the target expressions and repeated administration, implying a high safety bar. So in gene editing therapeutics, mRNA will encode for designer nuclease such as the zinc finger, the TALEN, as well as the Cas9 endonuclease. And this nuclease requires only a short duration and a transient expression from the mRNA in order to minimize the entire off-target effect. This picture here illustrates the forecast for the evolutions in the research market for the mRNA technology. So until 2025, the forecast is based on the mRNA vaccines for COVID-19. And the prophylactic vaccine in general are still expected to remain the cornerstone, which is also known as the main main applications to be used by the researchers in the market up to 2035. And with the therapeutic vaccines for cancer and mRNA therapeutics making increasing contributions towards the end of the forecast period. And this diagram here, whether the mRNA for prophylactic vaccines, therapeutic vaccines, or even with the therapeutic applications, this technology is relatively new and many are still in the preclinical stage as well. As you can see, it's drawn in the light green color. And in contrast, only a very small portions of the mRNA-based vaccines have been approved for the emergency use. So the picture here actually illustrates the predictions or even the market trends for the mRNA research from the 2021 to 2025. According to the report, the mRNA research market size is about 100 million in 2021, but it is forecasted that in 2025, it's going to reach over 260 million, indicating the fast growing of the mRNA research in this field. So here I am showing the two examples of the mRNA vaccines, one from the Moderna and one from the BioNTech. This sequence were the mRNA sequence for the S proteins and had already been published. Here I summarize some of the features of the mRNA designs. So in here, it includes the CAP1 in order to ensure the RNA is the mRNA that within the eukaryotic cells, and also an optimized 5' UTR and also the 3' UTR regions. The codon optimized signal peptides to send the spike proteins to the correct locations. And then the original codon optimized spike versions has actually two proline alternatives to ensure that the proteins appears in the correct form. And also the polyadenylate, the poly-A tails. Well, their poly-A tails is a bit different design. Their design is with the 31 poly-A plus a 10 nucleotide of the spacer, and also following to that will be plus another 71 of the poly-A tails. And then the codon optimizations adds a lot of the G's and also the C's to the mRNA. and also with the pseudo-uridines at the different sites are used to evade the immune system attacks, thereby can actually prolong the half-life of the mRNA. So now I am going to talk about some of the features that can be modified during the mRNA synthesis. But first, I would like to discuss about the codon optimization. It's actually very critical for mRNA synthesis. And at GenScript, our research team actually established the codon optimization tool that actually allows the clients for the researchers to optimize their sequence for the transcriptions. In here, I actually demonstrated 3 different articles from the prestigious publications, including the cell price, including the immunity, the molecular therapy. So the researchers from this who actually work in this mRNA research utilize our optimization tool in their projects. In brief, so all of these articles use our codon optimizations and then to improve the expression level after the transcriptions into the mRNA. And the codon optimization is also important because it helps to optimize the codon usage for the expressions host. And furthermore, this codon optimization is critical to optimize the GC-rich regions in order to modulate the mRNA immunogenicity. Another features would be during the mRNA synthesis, the T7 promoters that lies within these 5' UTR regions. The T7 promoter is actually commonly used for the IVT mRNA synthesis. And in this picture, you can see that the T7 promoter within the sequence. And you can see the Tata box within the promoters, and also the reverse transcriptions begins with the first base behind the Tata box, which is the G. For example, in here, this is the G here is a sequence for the RNA transcript. And this slide basically shows the mRNA-based modifications. As we're all familiar with, the ATCGs are usually the 4 main nucleotides that found in the DNA. And in RNA, there is a thymine, which is actually replaced the uracil. So replaced by the uracil. So the difference between the uracil and also the thymine is actually the lack of these methyl groups in the uracil. So here I show you a typical, this mRNA sequence as an example. So you could have realized that all of the uracils has been replaced by the pseudouridine, and following to that, these are a few of the modification types of the uracil that can be found in the mRNA synthesis. First, so let's take a look at the uridine. And so the picture here describes the wild-type uridines, and in the modifications of the uridine to the pseudo-uridines, the researcher will usually replace the C, the C here with the N. And then furthermore, the researchers make a slight modifications based on this pseudo-uridine with the additional of the methyl group at the N1 position. So this is known as the N1 methyl pseudo-uridine. And as for the cytidine, we can also observe a small modifications at the carbon position. So these are the main few modification types for the mRNA synthesis. The purpose of these modifications are to avoid the nuclease to degrade, and also in order to avoid the innate immunogenicity. A couple of things to take note of that. It is not advisable to make a huge modifications in the group, or else the T7 promoters might not be able to recognize during the reverse transcription. Additionally, the efficiency of the T7 promoter to each of the modifications may be varied, but won't be too significantly different. So the picture here indicates the comparisons between the natural mRNA and a modified mRNA. So in terms of the efficiency from a vaccine development project, As a result, it shows that the mRNA with the modifications has a better efficiency at the 95% compared to the natural mRNA, which is at about 48%. So now I am going to talk a bit of the mRNA capping. The panel A here indicates the original RNA without any capping, whereby in the panel B indicate the cap 0, of known as the 5' capping, in which the mRNA cap 2O methyltransferase will be added into the methyl group at the 2O positions of the first nucleotide adjacent to the cap structures at the 5' end of the RNA. So the mRNA with the cap 0 usually can help to avoid the recognitions by nucleic acid enzymes in the cells in order to prevent the mRNA from degradation. And furthermore, an additional of the methylations on the 2'O positions of the initiating nucleotide generates the cap 1. The cap 1 has been shown to have the importance in evading the cellular immune response in vivo. So the bar chart here indicate the luciferase protein expressions based on the mRNA with different capping positions. So usually with cap 0, they will still have some of the minimal expression level due to the mRNA has already been degraded. With the addition of the cap 1, the half-life will be prolonged, leading to a high expressions of the luciferase protein. So, and now I would like to talk about the poly-A strategy. Similarly, there are a couple of strategies that can be used in order to adding in the poly-A at the 3' end. The first strategy is known as the enzymatic polyadenylation after the transcription. As described, the poly-A tail can be added after the in vivo or the in vitro transcription. With the use of the poly-A polymerase, the poly-A can then be added. But the drawback of this approach is the user unable to control the number of the poly-A being added to the mRNA. Another approach is to design the plasmid backbone with the inclusions of the poly-A. So usually this design will follow with this enzyme which is known as the BSP-Q1, of which it will be used in order to linearize the plasmid for IVT mRNA synthesis downstream. So after the linearization, the in vitro transcriptions, as I said, will be taken place in the presence of the T7 promoters based on a template that consists of the poly-T sequence. And so with this strategy, we can actually ensure the number of the poly-A can be accurately controlled. The third strategy is through the PCR amplifications with the use of the two different primers, of which one of them consists of a T7 promoters at the 5' ends, and also another consists of the poly-T at the 3' end. So after the amplifications, the PCR products will be used as a template for IVT purpose. But the drawbacks of this approach is the generations of the byproducts. The number of the poly-A tails at the 3' end is very difficult to control and also being added. And therefore, the second approach is still the preference approach. So here I am going to show you a couple of the different designs with the poly-A tails. The first examples indicated 120 nucleotides of the poly-A at the 3' end in the plasmid backbone. And the second example, which was from the BioNTech, in which, as I mentioned, you may see the inclusions of the spacer in between the poly-A regions with a 30 poly-A in front and also a 71 poly-A at the back. Similarly, another mRNA designs from the researchers with the inclusions of the spacer in between the poly-A regions. And so now I would like to share with you a few case studies that conduct in-house to demonstrate the quality of our mRNA. The first case study that I would like to share was the GFP expression test using the mRNA that produced by GenScript in-house. As described, so this 1,000 base pair of the eGFP mRNA was produced based on our IVT production workflow and then transfected into the A549 cell line. The expressions level of this eGFP was investigated using the confocal imaging method. So as shown, you can see that the fluorescent intensity increased when the high amount of the mRNA being transfected into the cell, indicating that our mRNA quality is high enough that allow us to achieve a linear correlation between the intensity of the fluorescence and the amount used. And following to that, our production had also investigated the effectiveness of the different lengths of the poly-A in the mRNA. Again, by using the eGFP as an example, its mRNA comes with different lengths of the poly-A tails, including 41 to 80A, 42 to 60A, 44 to 40A. And also an mRNA was obtained from the competitor. And as you can see from this picture here, the GFP expressions in the A549 cells was actually higher when the mRNA consists of a longer length of the poly-A at 80 poly-A, indicating that the length of the poly-A tail improved the half-life of the mRNA in the cell. Furthermore, we also investigated of its usefulness by increasing the poly-A to 100A and even to the 120A. However, the data, which is not shown here, that there wasn't actually quite a big difference between 100A to 120A. So which is why majority of the researchers are sort of agree that 100 to 120 of the poly-A tail length is actually within the acceptable range. So now I would like to share with you about what and how GenScript can offer in the mRNA service. Okay, so we know about the applications of the mRNA, and then regarding to the production workflow, after receiving the mRNA template, our productions will have DNA synthesis and also with the plasmid cloning. And following to that, our productions will validate the clone plasmid by the Sanger sequencing in order to ensure the accuracy of the gene being cloned, and also through the restrictions enzymes in digestion analysis to ensure the synthesis accuracy. And then this plasmid will be linearized before transcribing the mRNA using the in vitro transcription. So the mRNA will then be purified either using a column-based approach, which at GenScript we call it a research grade of the mRNA, or to be further purified using the oligo-DT purification approach for preclinical grades, because it requires a higher purity when coming into the animal usage, or maybe to the animal testing. And finally, we move on to the quality control with the agarose gel electrophoresis approach. But in addition to the agarose gel electrophoresis, we also have different types of the QC available in order to test and also to ensure the quality of the mRNA being delivered to our clients. Here are some of the features that GenScript can do and what services we can offer. Starting from the scale, we can offer a wide range of a different quantity from a micrograms level into milligrams level. So the highest that we can do now is about 200 milligrams. As for the 5' capping options, we can now do whether it's uncapped, cap 0, or cap 1. And for the poly-A tail options, we can help with 100A poly-A tails, but if there is a request up to 120A, yes, we can also do that as well. And with the modified options available, we have different types of the pseudouridines available for the modifications. But we usually will recommend the N1 methyl pseudouridines simply because our productions obtain and also recommend that this will provide a best outcome based on with these modifications. And moving forward to the purification approach, as we mentioned, we have a couple of a purification approach available depending on the requirement of the mRNA quality. So this includes a column-based purification approach that in order to achieve a research-grade mRNA, and also to further purify using the oligo-DT approach in order to achieve a higher purity of the mRNA for preclinical grades, preclinical studies. And for QC package, we have a sort of a different type. We have a research grade of the QC availables and also a preclinical grade of the QC available, which I will further discuss in the next slide. So this one is the QC specifications. So as I mentioned earlier, we have a different QC package available for research grade and also the preclinical grade. And the quality of the mRNA will be assessed from 4 different perspectives, including the identification, the purity, the impurities that present in the samples or in the products, as well as the safety. And these are the items that we can that we will actually investigate, and also followed by a different approach that in order to achieve the specifications that the specification that we set before the release, so that we can ensure the quality of the high quality of the mRNA can be delivered to the researcher. So before I wrap up my presentation today, I would like to highlight the advantage of the working with GenScript on an mRNA. With our in-house capability, GenScript can help with the synthesis from the gene to the IVT step in order to minimize the need to source the materials from multiple vendors, and also to reducing the risk of supply. And most importantly, GenScript has more than about 19 years of experience in producing the customized nucleic acid for the biopharma, as well as for the academia and industry. So now I would like to wrap up my presentations again to present you a couple more of the services and products for therapeutic purpose. Here, as you can see from the slide here, GenScript actually offers a wide range of the services and products, include the molecular biology that consists of gene synthesis as well as the plasmid prep, the IVT mRNA service, which I have already mentioned, the oligosynthesis approach, and also the CRISPR-Cas9 genome editing. So as we all know that moving towards the gene and cell therapy, the researchers have also started to investigate the feasibility of using the CRISPR for gene and cell therapy purpose. And with that reason, GenScript also have established our capability to provide the materials from research grade up to GMP level. For the peptide synthesis, at GenScript, we also provide a customized peptide synthesis that can with a different grade, including the research grade as well as the cGMP level. And we also can help with the protein expressions as well as the antibody generation service. This is this graph basically is to demonstrate our capability in providing different grade of the plasmid prep. So a lot of the time, the researchers have been relying on our vendors to provide a research grade of the plasmid. However, especially when coming into the therapeutical research, at different stage, you will need different grade of the plasmid for your research. For example, when you're still at the very beginning stage, during the R&D stage, you will probably just need a research grade plasmid to understand about your gene of interest. But when coming into the transfections, a lot of the time the researchers might facing some of the issue like transfects not enough of the plasmid into the cell. So we have this industrial grade of the plasmid that can actually guarantee the supercoid while minimizing the endotoxins. By doing that, we can actually ensure the cell viability and also to increase the transfections of the plasmid into the cell. We also have a preclinical grade of the plasmid that is suitable for the animal study, especially when for the gene and cell therapy purpose. And we also have the plasmid grade, which is the pro plasmid and the GMP plasmid that helps the researchers for IND filing human clinical trials, and as well as the commercialization purpose. Now, as for CRISPR, as I mentioned, we offer a wide ranges of services and products. So we have this, this plasmid as well as a vector-based CRISPR for the gene and cell therapy purpose. And recently, a lot of researchers have been trying to use the RNP, which is a ribonucleoproteins products for the genome editing. The material includes the single-strand format of the guide RNA, readily used Cas9 proteins, and also a donor template in a single-strand DNA or a double-strand DNA format in order to generate, for example, the CAR T cells for gene and cell therapy purpose. And we also have a CRISPR libraries for screening in order to understand the genes that are related to the diseases. And we also have a team of the experts in CRISPR that help the researcher to generate the cell lines, whether it's in a knockout or a knock-in. Also, we can help the researcher to do the microbial editing service using the CRISPR on the E. coli. This is just a summary of what I mentioned. For CRISPR technologies, we can actually provide the different grades of the materials ranging from the research grade up to GMP level in order to fulfill the demands of the research need from the research grade up to the clinical trial. So again, this is just a summary to highlight our capability in a to produce the GMP grade of the sgRNA, ssDNA, as well as the double-strand DNA. And for the Cas9 endonuclease, we also can provide the research grade and also a GMP grade of the Cas9 endonuclease for the in order to produce to conduct the genome editing purpose. But here, I will just stop here and I thank you for your attention and I welcome any questions. Thank you.